So next we will hear from Dr. Martin Grosell. Dr. Martin Grosell is a Mahi Professor of Ichthyology at the University of Miami's Rosenstiel School of Marine and Atmospheric Sciences. He is also the director of the GOMRI Consortia Recover.
Thank you so much for the introduction. I am the director of the Recover Consortium funded by GOMRI. And let's see if I can advance my slides here. There we go. The Recover Consortium, as I mentioned, was made possible by the Gulf of Mexico Research Initiative, or GOMRI. And I want to start by extending my gratitude to GOMRI for allowing what I think is a unique opportunity to look at the impact of environmental disaster all the way from the molecular level in marine organisms all the way up to population level, spanning cell function, organ function, performance of intact or whole animals, how they interact, and ultimately what that means for population level impacts. We have focused mainly on two species in this consortium, the Mahi, which is a representative of the pelagic community in the Gulf of Mexico and in fact across the world, and also the red drum, which is a common representative for the coastal areas in the Gulf of Mexico. And GOMRI allowed us to form this consortium, which again brought together a very wide range of sub-disciplines and even disciplines to look at the impact of oil spills on marine fish. And as I mentioned, this is an opportunity that I would have never gotten otherwise. And I think some of my most memorable moments have been working in these larger groups, whether it's just say a dozen people or even bigger, where it's been very clear that the intellect and experience of a wide range of scientists, when combined, have produced something that's much, much greater and more impactful than just the sum of its parts. And I cannot express enough how important that has been to me in my career, and I think also to some of the discoveries that I'm going to be discussing next. Working on pelagic fish is not easy. We have chosen Mahi because we were able to successfully breed them in captivity and in fact raise them in captivity also at the experimental hatchery, which is what you see on the right image here, at the University of Miami's Rosenstiel School of Marine and Atmospheric Sciences, or RASMUS. The RASMUS campus is located just across the street, and both facilities on Virginia Key here, just a few miles offshore from downtown Miami. Culturing Mahi taught us a lot. It taught us that the embryonic stage is very short. The embryos last for about 36 hours and hatch. The larvae have the highest growth rate of any marine fish examined to date. They grow up to 30% per day. They reach young adulthood or sexual maturity at about 90 days and are able to spawn. And in fact, we have closed this life cycle in captivity several times. This has allowed us access to all life stages of Mahi, I wouldn't say daily, but very frequently, and allowed the research that we've conducted at RASMUS at the University of Miami. And I'll be sharing some of that. One of the first things that we realized was that crude oil toxicity is a multi-organ syndrome. Many factors in an animal's impact when exposed to crude oil. They include cholesterol biosynthesis, which we believe is central to many other effects, mitochondrial function or dysfunction, stress response has been mentioned already, compromised immune function. And what we've learned, which is sort of fairly novel, is that sensory systems, including vision, olfaction, and also the lateral line I'll be talking about, are impacted by crude oil contamination. And then finally, and no less significantly, heart function is severely impacted by oil exposure. I don't have time to talk about all this, but I'll focus on a few examples, the first being vision. And you can actually perform an eye test or a visual acuity test on larval fish. What you see here is a Mahi. It's about 20 days old, and it's placed in a cylinder around which we are moving black and white bars, so these contrasts between black and white. And you'll see how this fish faithfully follows these contrasts. And what we can do in this system is we can increase the speed of rotation until the fish is no longer able to see the contrast, where basically these alternating dark and light fields become a blur and the fish stops tracking them. And we do that, we get a measure of the visual acuity or the ability of these animals to see their environment. And if you look at the white bars here, this is the development of visual acuity or eyesight in larval Mahi from 7 to 10 days post-hatch. And you can see that as they age and mature, they become better and better at seeing things. What you can also see in the filled bars here, the gray bars, is that animals that have been exposed to an exceedingly low concentrations of polycyclic aromatic hydrocarbons, which is a common component of the crude oil, they have much lower visual acuity. So essentially, eye development and eye function is impacted by oil exposure. And of course, as you can imagine, being able to see is critical for capture and prey and other functions that are important for survival in the wild. Many people are not aware of the 6 stents that fish have. It's comprised of neuromasts, which are basically bundles of sensory cells that are sitting right under the skin. From these sensory cells, sense hairs extend into the water or into a fluid-filled canal that's right under the skin, but connected to the outside. And essentially, any displacement of movement in the water will displace these sense hairs, signal the sensory cells to then communicate the observation to the brain or the central nervous system. What you're looking at on the right here is a larval fish 48 hours post-hatch, and the green stains here are individual neuromasts. You can see the individual sensory cells and their hairs right here. Now, this system is important for detecting movement in water, for acceleration, auditory stimuli, basically anything that's going on in the surrounding environment. And what typically happens in a larval fish in response to a stimuli is the C shape and then an extension of the tail that propels the fish forward and away from the source of the stimuli, which could be a predator. Now, we've looked at the startle responses, we call this, in zebrafish larvae that were exposed to the Deepwater Horizon crude oil. And what we're looking at here first is animals that are not being stimulated. An automated video tracking system basically detects how much they're moving. And you can see that these short red lines here are basically representing their movement for a second within these wells prior to stimulation. In the right image here, you're looking at the movement during 1 second of stimulation. And you can see that these lines are much greater, showing that the fish are responding to the stimuli by the startle response that propels them away from where they were at the time of the stimulus. We can quantify that, and we've done that for oil-exposed and non-exposed fish. And what you're seeing in the black bar here is the distance traveled for a second during stimuli in control, healthy fish. And in the gray bar here, the corresponding distance traveled by fish embryos or fish larvae that have been exposed for oil for 24 hours prior to this test. So this clearly has implications for, well, it could have implications for predator avoidance in the natural environment. We have seen impact to other sensory systems as well, but in the interest of time, I'm going to move on to impacts on the heart. What you're looking at here is a young adult Mahi. It was bred and raised at the experimental hatchery at the University of Miami, and it's in a swim tunnel. And in this swim tunnel, we can increase the velocity of the water and thereby the swim speed of the fish, being completely, it's basically the same as a treadmill for humans. And we can keep increasing the speeds until we get a measure of the maximal swim performance of these animals. We refer to that as UCRIT. And when we do that, we see that the maximal swim performance is reduced when they've been exposed to even very low levels of crude oil after just 24 hours. These are concentrations that were well within what was measured in the Gulf of Mexico. In this system, we can also measure the oxygen uptake of the animal, which is an indicator of the energy consumption. And if you're looking at the oxygen uptake capacity here, so basically the ability to extract oxygen from the water, you can see that it's greatly reduced under the same conditions. And we believe that this reduction in oxygen uptake capacity is what explains the reduced swim performance. Now, when you witness reduced oxygen uptake and swim performance in fish, you think of 3 possible explanations. One is reduced uptake at the gill of oxygen. We've looked at the gills, they're fine, they should not present a problem for oxygen uptake. Another is the conversion of oxygen to energy. This is occurring in the mitochondria in the cells, could be muscle cells that allow for swimming. We do see some impact on mitochondrial function. It's not consistent, and I don't think it explains the reduced swim performance. It could be part of the story, but we see massive impacts on the heart. And of course, the heart is important here because it drives blood through the gills where it's oxygenated, and then to the tissues where it's consumed, and then back around. Now, we've looked at the heart in more detail, and what you're seeing in this video here is an isolated heart cell from a Mahi. At the time of isolation, this Mahi was healthy, and we can stimulate the heart cell electrically, as you can see here, and when we do that, it contracts. This is what's happening to heart cells in an intact heart, and that's what actually drives the heart to pump blood. We can then perfuse this cell with salines containing contaminants from crude oil collected in the Gulf of Mexico. And when we do that, these heart cells are being stimulated electrically, and when they are, they shorten. And what you're seeing here is that the ability of these cells to shorten is reduced after oil exposure. I mentioned that occurs after seconds. We've also looked at the intact heart. This is heart surgery in Mahi. What you're looking at here is the ventricle, so that's part of the heart. This is part of the heart that pumps the blood. And you're looking at the blood vessel or the artery that leads the blood from the heart to the gill. And what we have here is a flow probe that detects movement of blood cells through this artery. And when we look at that, we can see that the heart rate here is not impacted by oil exposure. The open bar is the control, the hatch bar is the oil exposed animal. But when we look at stroke volume, so this is the amount of blood that's leaving the heart for every contraction, we see that it's greatly reduced after exposure to pretty low concentrations of polycyclic aromatic hydrocarbons. And of course, the heart rate and the stroke volume is what dictates the cardiac output. And this is really what matters to the fish. This is the amount of blood that's pumped from the heart to the gills and the rest of the animal. And we see that's greatly reduced also. And we believe that's explained by this reduced ability of the heart cells to shorten. And we believe that that explains why these fish swim less well and take up less oxygen when they've been exposed to oil. So I've shown you that we have impact on sensory systems and heart function. You have to trust me, we have impact on these systems also. And the big question now becomes, these animals that we show these effects on, they were in the lab and they were healthy at the time of these measurements. What happens in the wild where food is not provided to these animals and where there actually are predators that they have to deal with? And to answer that question, we combined exposure of wild fish with a Popup archival satellite tag system or experiment. We caught and tagged animals, maintained them in these 5-ton tanks on board our research vessel. Some were exposed to oil, some were not, for 24 hours or overnight until they were released the next day. I'm just going to show you a brief video here. The fish were collected from sports fishing vessels, brought to the research vessel, tagged very quickly, introduced into the tanks where they're exposed overnight, and then released the next day to do their thing in the Gulf of Mexico. Now, so far, we have tagged and released 69 Mahi over the last 5 years. And these are the tracks that we've collected from these animals once we record or receive the tags or the data from these tags. And a couple of things I want to point out here. We actually have fish moving between the Gulf of Mexico and the Atlantic and back. And what that means is that if you're assessing the impact of a disaster in the Gulf of Mexico, you cannot avoid the Atlantic. You can't assume that the effects are limited or restricted to the Gulf of Mexico. What we've also learned is that Mahi travel up to 100 kilometers a day with these tags. They reside mainly in the upper water column, but they will be deeper at night. They display a very tight temperature preference. They prefer 28 degrees Celsius very faithfully, and they seek that temperature by following currents or eddies, and also by adjusting their depth to achieve that temperature. We've also learned from the tags that Mahi spawn at night, chiefly at the new moon phase. Females spawn on average once weekly, which is less frequent than what we see in captivity. The males can spawn more frequently. And what this points to is that Mahi are actually highly fecund fish. Last year, we undertook a cruise into the Gulf of Mexico. This is the cruise track here, and the little dots are all the sites where we caught, tagged, and released Mahi. Half of them oil exposed and half of them exposed to controlled conditions. This is brand new data. It's still being analyzed. But I can share with you this plot here that shows survival probability of wild and controlled fish that have not been oil exposed in blue, and survival probability of oil exposed animals here in the orange. We're just showing the data here for the first 7 days because that's where the biggest differences are occurring. And what we're seeing is here that the oil exposed Mahi have twice the likelihood of dying within the first week after release compared to the controls. So there clearly are impacts of oil exposure also on wild fish. This is a sample trace of a Mahi. What you're looking at here is normal Mahi behavior in the first 3 days. The dark bars here represent nighttime, the white bars daytime. Slightly deeper at night, slightly shallower during the day. And then at this point here is a predation event. And we know this because there's no more light being absorbed or recorded by the tag, meaning that the tag has been ingested. And you can also see that the behavior of the tag changes dramatically after this event. We don't know who ate this Mahi. We know it's not a mammal because of the temperature recordings. We're suspecting based on the behavior that it's a 6-gill shark. This occurred in the Atlantic, but we don't know for sure. What we know for sure is that 75% of the tag deployments ended with confirmed predation, and that is twice as likely for the first week after oil exposure in Mahi compared to controls. We also know that predation events occur mainly or almost exclusively at dusk and dawn, and this is consistent with the fact that Mahi have very well developed eyesight for high light conditions, but they see poorly at night, which is in contrast to billfishes, sharks, and many of their other predators. So as I mentioned, impairment of sensory systems and heart function probably leads to reduced survival in the wild fish, which sort of supports the information that Tracy Sutton shared just a few minutes ago. In terms of outreach, I'll be brief on this. Some of my favorite moments from outreach have been the traditional sort of one-on-one or one-to-group type outreach events. In this case here, we have one of our former postdocs literally lifting this young girl up to a higher appreciation and understanding of marine science and oil spill science. We've communicated with the sports fishing communities and boating communities at boat shows and through seminars, and in this case in an article in the Florida Sports Fishing magazine. But perhaps most importantly, I think from an outreach perspective, we have developed an app. It's available for free in the App Store that targets elementary and middle school kids as well as high school and university students and allow them to perform virtual experiments illustrated by real data collected by Recover scientists and also narrated by our scientists. And this has greatly expanded our reach in terms of outreach. We have reached more than 1,200 students with this app, and we're still adding to that. We have reached 65 schools in 28 states, and in fact, one outside the states also. And that sort of concludes what I had to say today. I just want to draw your attention to our website where there's more information, and also our social media platforms and my email address should you have any questions that we don't have time to address later today. Thank you.
Thank you, Dr. Grosell. As he mentioned, if you have questions, we will take, hopefully we'll have time for questions after we have 2 more speakers. So if you have a question, please put it in the chat box on Zoom or in the comment section on our Facebook Live portion of our webinar.